Hello, and welcome to the Connect Science Seminars in Oncology series of the Dana Farber Cancer Institute. These seminars are held every Tuesday and Thursday at 12 noon US Eastern Standard Time. And please go to the website shown on the screen for a list of forthcoming talks and recorded past presentations. So one housekeeping announcement before I introduce the speaker is not to submit your questions in the chat, but to the Q&A session. So today I have the great pleasure and honor of introducing Dr. Howard Chang, who is the Virginia and D.K. Ludwig Professor of Cancer Research, Stanford University, and Professor of Dermatology and Genetics and investigator, Howard Hughes Medical Institute. Howard obtained his MD from Harvard University and his PhD from MIT at the lab of David Baltimore. His postdoc fellowship was in Patrick Brown's lab at Stanford. Howard has made a number of seminal discoveries in the field of long non-coding RNAs and in their roles in the regulation of gene expression, with a recent focus on their effects on chromatin and their roles in chromatin regulation in normal and disease states such as cancer. His lab has also developed new technologies for studying the non-coding genome in a genome-wide fashion. He has received innumerable prizes and honors and is a member of the National Academy of Sciences and Medicine. So therefore, thank you so much for joining us, and I really look forward to your talk. Howard?
Thank you, Ronnie, for this very kind introduction. It's really a pleasure to reconnect with many friends and colleagues at Dana Farber and Harvard Medical School. Let me move my slide forward. My lab has been fascinated by the non-coding genome. When we think about gene expression, we often think about disease-causing genes. But this slide reminds us that connected to every gene are a series of DNA switches that decide when and where genes turn on and off. And these non-coding DNA elements are actually the binding sites of regulatory proteins such as transcription factors and or regulatory RNAs. And in fact, the picture in the human genome looks more like the bottom. Just 2% is protein coding. And the vast majority, 98% of the real estate, is actually this non-coding genome. We also know that this is where most of the inherited variants associated with human disease will lie. And so what I thought I'd do today is to talk about some principles that we've learned over the last few years by focusing on an oncogenic lincRNA locus. I will first talk about its regulation in the chromosomal context. In the second part of my talk, I will talk about our discovery about this same locus being amplified in extrachromosomal DNA. And the final part, I will talk about some new lessons, new ideas about how gene expression may operate in the ecDNA context, extrachromosomal DNA context. So let me start by sort of reminding you that in every human cell, 2 meters of DNA is packed into a 10-micron nucleus. And therefore, most of your DNA is highly compacted and not accessible, except for the active DNA elements that's being read by the cell's machinery. Therefore, simply finding these accessible elements can teach you a lot about the software program any particular cell is actually running. So based on that idea, many years ago, my colleague Will Greenleaf and I introduced this technology called assay of transposed accessible chromatin, ATAC-seq. In fact, that was developed by Jason Boyne-Rostro, a new faculty member now in the Harvard community. And so this method uses an enzyme that copies and pastes DNA called transposase. And we load it up with sequencing adapters that can go into our sequencing machine. When this enzyme interacts with eukaryotic chromatin, it can only copy and paste into the open sites. And therefore, in a single step, you selectively and covalently tag the regions of interest, making it possible for you to then amplify and sequence the open chromatin landscape. This method led to a pretty amazing million-fold improvement in the sensitivity and a hundred-fold improvement in the speed of mapping active DNA elements. And so let me show you some primary data. This is what the data will look like. So in the first row, so we're looking at genomic tracks. On the x-axis are basically genes and DNA elements. On the y-axis, each peak indicates a level of accessibility. And so the height indicates these are the active elements. The first row is a prior gold standard, DNase hypersensitivity, which used 10 million cells. The second row was the first version of ATAC-seq, which was done with 50,000 cells. And then a few years ago, we achieved the ultimate resolution, single cell ATAC-seq. That's the third row. This is a summation of several hundred single cells. You can see that these tracks look very similar, but now you can actually zoom in. And so now every row at the bottom is a single cell. There are 254 single cells here. At every position, you can see either 0, 1, or 2 reads because this is a diploid genome. So in this way, this analog signal can turn into digital information. Armed with this very powerful technology, we're able to access and study certain archival samples that are previously out of reach. And one of the exciting applications was applying ATAC-seq to samples from the Cancer Genome Atlas, TCGA. I know this is a topic that's very familiar to the audience. In fact, a lot of the foundational work for TCGA was done at the Dana Farber. And so let me just briefly say that this is I think one of the flagship projects in the cancer research community of the last decade. Investigators all across the country have collected many kinds of cancers for very in-depth genomic analyses. But at the time, we didn't actually have insights into its chromatin landscape and or gene regulation. And so using ATAC-seq, Ryan Corses, a very talented former postdoc, was able to analyze the open chromatin landscape in 23 human cancer types, which are shown here. These include some of the most common and deadly human cancers, ranging from glioblastoma, GBM, lung cancer, breast cancer, and so on and so forth. He studied 410 tumors, and we identified over 500,000 active DNA elements, which are putative enhancers and other kinds of regulatory elements that activate and control gene expression. Of note, when we compare to the landscape of normal gene expression in, for example, normal tissues, in epigenomic roadmap, ENCODE, and other studies, nearly half of these elements are actually not seen in normal biology. They're only exercised during the pathology of cancer. Let me show you an example of a very important oncogene, and this is the MYC locus. So on the right, I'm again showing you genomic tracks. The first 5 rows shown in orange are colon cancer samples. The bottom 5 rows are actually kidney cancer samples shown in purple. The MYC locus, if you can see my cursor, it's right at the bottom here. So this oncogene is actually active. It's accessible in all these samples, but you can see that different cancers use different regulatory elements. Colon cancer, mostly using elements to the 5' end, and kidney cancer, mostly using elements to the 3' end. So same oncogene, different oncogene switches. Moreover, this kind of map provided a lot of insights into inherited variants that are associated with cancer risk. Cancer geneticists have been mapping different families that have an increased chance of getting different kinds of cancers. And most of the associated elements are actually in the non-coding genome. So for example, there's a colon cancer risk in the 5' locus of MYC gene. And this turned out to be a colon cancer specific enhancer shown in this map. And conversely, kidney cancer risk is associated with the different single nucleotide polymorphism, a SNP, which is actually on the 3' end. And this maps right to a kidney cancer specific enhancer that's actually in the PVT1 link RNA locus. And that is going to be the focus of a lot of our discussion today. So this kind of map also, of course, extends genome-wide and is very informative for many kinds of cancer. So let's talk about this MYC and PVT1 locus. My lab has been interested in PVT1 even before this work for several years, because PVT1 is probably the first linked long non-coding RNA gene implicated by cancer genetics back even in the 1980s. So PVT1 stands for plasmacytoma variant transcript 1. It's actually the breakpoint of a translocation in Burkitt's lymphoma that led historically to the recognition of MYC as an oncogene. It is co-amplified with MYC in many cancers. But despite the fact that it seems to be somehow has this oncogenic flavor, there are frequent mutations and deletions in this locus. So you might wonder, if this is an important cancer-driving gene, why does cancer want to mutate it? So hopefully towards the later part of the talk, this rationale, the mechanism, the driving force might become more clear. MYC is, of course, a powerful oncogene that's overexpressed in half of human cancers that can drive all the growth, the key hallmarks of cancer important for cell growth. So again, just to clarify the locus organization, MYC and PVT1 are two genes transcribed in the same direction. They're 53 kb apart, and they're promoters, and this PVT1 transcript is a very long transcriptional unit. My lab has had a long-standing interest in understanding and discovering a link RNA. And a few years ago, we teamed up with the lab of Jonathan Weissman to perform a genome-wide screen to understand cell type essentiality for long non-coding RNA genes. We used CRISPR interference, which is essentially you repurpose CRISPR-Cas protein to use a catalytically dead version, dead Cas9, as a DNA binding platform. This is fused to the CRAB repressor domain, so we can actually target this construct to the promoters of many different link RNAs, silence their transcription, and look at the impact on gene expression and or cell growth. We developed 10 guide RNAs to each to over 16,000 human link RNA loci, and we conducted a genetic screen, a pool proliferation screen in 7 different cell types. And this analysis revealed approximately 500 essential link RNAs for cell growth or survival. Strikingly, most of them were only essential for one cell type only. So there's a high degree of cell type specificity. Now, coming out of that same screen, we're also kind of the opposite phenotype. Link RNAs that, when silenced, make the cell grow better. So these are putative tumor suppressor link RNAs. And the #1 hit in the entire transcriptome was the PVT1 promoter. So this is an example of such data. So you're looking at the frequency of the guide RNA, and over basically 20 days of cell proliferation, you can see that the frequency, the allele of the PVT1 silencing guide RNA would increase in the population, as seen in, for example, these breast cancer cells, GBM, another breast cancer line, and so on and so forth. Okay, so Sungwoo Cho, a very talented postdoc, basically decided to study the PVT1 link RNA, PVT1 promoter. And this is an in vivo xenograft assay. And these cancer cells are harboring luciferase, so we can track their growth. So in this xenograft assay, the left tumor cells, or breast cancer tumor cells, were silencing PVT1 promoter. The right was a control guide RNA. You can hopefully see then that basically every time the dot on the left is more bright, there's a growth advantage in vivo. And in vitro head-to-head cell competition assays, we find that after about 10 days, the PVT1 promoter targeting cells would always outgrow the competition. And so there's a very strong growth advantage. Sungwoo went on to then conduct RNA-seq to understand how PVT1 would have this kind of tumor suppressor effect. And so this is a RNA-seq this is a volcano plot on the left. So the CRISPR-I targeting PVT1, of course, and we're gratified to find that one of the most prominently downregulated transcripts was in fact PVT1 itself. And the most upregulated transcript in the entire transcriptome was MYC, because oncogene is 53 kb away from the PVT1 promoter. This antagonistic relationship was also verified on the protein level. If we silence the PVT1 promoter, MYC RNA level and protein level would also increase. So we can think about this relationship like this seesaw, right? So PVT1 and MYC, they're connected next to each other, but when PVT1 is down, MYC is up. And also in experience, I'm going to show you the reverse is also true. We're very excited about this result, and because my lab had done a lot of work on long non-coding RNAs. But after many experiments, we discovered that it was not the PVT1 RNA that was responsible for this phenotype. In brief, other kinds of experiments that would get rid of the PVT1 RNA didn't have the same growth promoting effect, was actually specifically silencing the PVT1 promoter, the active transcription. Okay. And this led us then to really think about a different kind of mechanism, a DNA-based mechanism rather than RNA that might be responsible for this effect. And so to that end, we turned to another technology that was developed in the lab called an enhancer connectome using a method called HiChip. This is a method that gives you both, identifies both the enhancer strength and enhancer targets in the same assay. Briefly involves essentially making a proximity ligation in situ, essentially a high-C to connect DNA elements that might be coming together in proximity in the living cell, followed by an IP enrichment, a chip assay for a protein mark, in this case, the enhancer-associated mark, histone H3, lysine 27 acetylation. And so this method has several technical sort of advantages, which we like. Okay, so we can actually then map out all the active enhancers and the promoter contacts, and that proved to be very informative. So for example here, on the top, I'm showing you something called a virtual 4C view. We're looking from the viewpoint of the MYC gene. Each peak here on the top is an active enhancer that is now touching the MYC gene. In gray is the wild type control situation, and in red is what happens when you shut down the PVT1 promoter. And so what we discovered is that there are basically 5 intragenic enhancers that normally, in the intron of PVT1, that normally activate the PVT1 promoter. But if the PVT1 promoter were shut down, these enhancers are still active, but they reach over and activate MYC instead. Okay, so this PVT1 promoter is really like a boundary element that prevents the enhancers from spilling over. A very strong prediction of this model is that the PVT1 promoter should then act in cis, that is on sequences on the same chromosome, but not across different chromosomes, not in trans. And so at the time, we were very happy to find that in a separate study in the lab, we were studying monoallelic gene regulation, and PVT1 happened to be one such gene, allowing us to kind of test this idea. So let me just briefly explain that obviously in every human cell, you have copies of genes, one from mom and one from dad. These are 2 different alleles. In most cases, both alleles can express and fire equally, but there are other interesting situations when only one or the other allele is allowed to transcribe. And Jin Xu in the lab was conducting a screen for monoallelic regulation, random monoallelic regulation, and PVT1 happened to be one such gene. So in this analysis, where you're looking at an F1 hybrid cell, these 2 chromosomes are marked with a very dense single nucleotide polymorphism. So in each track, I'm going to show you 2 colors, one color from one allele and the blue color for the other allele. And so, for example, in embryonic stem cells, looking at the MYC PVT1 locus, you can see that everything is the same for the chromatin accessibility, RNA expression, promoter activity, and heterochromatin marks on the 2 alleles. But when embryonic stem cells differentiate into near precursor cells, we discover that, in fact, there is now monoallelic regulation. It is random. It can either one of the 2 copies of the PVT1 will transcribe, and then that choice is remembered in all its daughter cells. Okay, so this is actually a clonal event. So you can see now that in ataxia data, in this particular line, the blue allele is active. PVT1 is transcribed. The blue allele has the active histone promoter mark, H3K4 trimethylation, and the other allele, the red allele, is now silenced by heterochromatin as H3K9 trimethylation, shown here. And then when that happens, the allele that has active PVT1 has lower level of MYC expression. The other allele has higher expression. Okay, and so this really shows then that as we look across many clones, each dot here is a different clone that's made a different monoallelic choice, that the more copies of PVT1 that's active, the lower the MYC mRNA level basically steps down. Moreover, looking at the allelic choice, whenever the allele bias for PVT1 is increasing that allele, the MYC expression from the same chromosome then goes down. And that's why there's this negative slope. Okay, so this suggests then really that PVT1 regulates MYC in cis in the chromosomal context. And so remembering our seesaw, right, MYC and PVT1 going up and down. Another way to remember this is that to play seesaw, the two players have to be on the same plank. So for this regulation to happen, PVT1 and MYC DNA elements have to be on the same chromosome. So to summarize then, this initial understanding of the MYC PVT1 locus in the chromosomal context, we recognize then that the link RNA promoter is actually, the link RNA gene could be really more fruitfully understood as a series of DNA regulatory elements. And the link RNA promoter is actually a tumor suppressor DNA boundary element. The cell growth competition that we see at the phenotypic level reflects a molecular competition at the DNA element level, where these enhancers in the PVT1 locus are competing between to go to either the PVT1 promoter or the MYC promoter. And the PVT1 promoter actually has to capture these enhancers to prevent them from spilling over. And other work that I don't have time to show you found that the PVT1 promoter is actually the hotspot for mutations, loss of function mutations in human cancer samples, and that an experimental mutation, a PVT1 promoter, even as small as a 6-base deletion, is able to abrogate its function and give the cell a growth advantage. So far, I've kind of focused on the PVT1 promoter as a putative tumor suppressor. But this model also predicts that perhaps the enhancers will also should be oncogenic, and maybe they'll be amplified. And so we have actually an opportunity to look at this idea in actually a really interesting context. What I've shown you earlier are basically ATAC-seq data that look like individual peaks, look like active DNA elements. But sometimes we see data like this, where we see the very broad swath of strong chromatin accessibility across, for example, like a megabase size here, such as seen for the MYC PVT1 locus in this prostate cancer cell, or let's say the EGFR locus in this glioblastoma cell. And we have, this is a very unusual pattern of DNA accessibility. We have so many reads in such regions that we can actually stitch them back together. And we actually found, in collaboration with Paul Michal's group and Vineet Bapna, that in fact, these are actually circular extrachromosome DNAs that encompass the oncogene. Okay, so the two ends here will actually be joined together like this. And so this is actually like an oncogene plasmid. This kind of an idea obviously needs a number of sort of validation, including long read sequencing, optical mapping. But I think the most convincing sort of strategy is actually direct visualization. So here I'm showing you scanning electron micrographs of a metaphase spread. These big structures in the middle here, these are chromosomes. You can see that they're DAPI positive. Then you see additional little dots that are also DNA. And when you zoom in, each one of those dots is actually a circle, and that's the circular extrachromosomal DNA. In a previous nomenclature, also called double minute chromosomes. We're going to talk about that term later, but they're mostly actually singletons, so it's a bit of a misnomer in the past. Okay. And so we were very intrigued by this finding. And in fact, there's a kind of resurgence of interest in ecDNAs, extrachromosome DNAs in cancer. So let me just summarize to say that they're large and they span from 100 kb to a megabase. In cancer, they almost always harbor oncogenes. And so on the left is a map looking at either TCGA data in red or a second data set in blue, looking at the cargoes in the ecDNA. As you can see that these are the who's of cancer-causing genes, including genes like epidermal growth factor, receptor EGFR, MYC, we just talked about, cyclin D1, KRAS, CDK4, HER2, so on and so forth. They're actually quite prevalent in human cancer. Half of human cancer types, a quarter of human cancer samples harbor ecDNAs. And they actually can cause really high degree of copy number and sequence variation. And that is because of their unusual mode of replication and transmission. So they actually do replicate, but as you can appreciate that extrachromosome DNA as a circle, it has no centromeres and it has no telomeres. And therefore, when a cancer cell divides, there's nothing that makes sure that the two daughter cells will inherit equal copies of ecDNA. All the copies can go to one daughter cell and the other daughter cell gets nothing. So you can really quickly basically jackpot and create a very high degree of oncogene copy number, selection, and then rapidly sort of jack up the gene expression level. And indeed, recent work together with Rover Hawk and other colleagues show that ecDNA amplification is strongly associated with worse prognosis and increased risk of death compared to other kinds of amplifications, including complex rearrangements and simple straight-up amplifications. So we're intrigued to discover sort of the mechanism of how the ecDNA could have these effects. And we found that ecDNAs are associated with very high levels of oncogene expression. On the left, I'm showing you a ranked sort of list of all the transcripts from the cell that carries the circular amplification ecDNA. The red dots are the genes on the ecDNA amplicon. You can see that they're ranked from the highest to the lowest. And you can see that, for example, MYC that was on this ecDNA is now expressed like 1,000-fold higher. So they're way higher expressed than what is normally possible. On the right-hand side, we're showing an analysis of TCGA samples. Now we have both chromatin and matched RNA. Those samples that carry the circular amplicon have much higher level of expression of the RNA compared to linear amplification, even though at the copy number level, they're amplified to the same level. So this really suggests that somehow you get more expression from the circular ecDNA. This idea was kind of directly tested from this experiment from Paul Michel's lab. In this ecDNA line, we identified a mutation, a SNP, that distinguished the ecDNA allele from the chromosomal allele. So the ecDNA allele is the major allele. So again, using this allelic analysis, you can walk down the chromosome, and you can see that on the whole genome sequencing, the ecDNA is present on, let's say, 100 copies versus 2 copies of the chromosome. But in the RNA-seq, the ecDNA-derived mRNA is now present at 400 copies instead of the 2 copies compared on the chromosomal DNA. So it implies really that pound for pound, the ecDNA is driving much higher levels of oncogene expression. Now at this point, you're probably thinking that, all right, Howard, you have high oncogene expression, but you also have high copy number. How are you really going to prove that at a single molecule level, each one of the ecDNAs is supporting more transcription? And this really needs, I think, an in situ and imaging method. So a few years ago, we developed this method called ATAC-seq to image the accessible genome. I told you that ATAC-seq operates by using a transposase to basically tag the accessible sites with DNA adapters. In ATAC-seq, we append the ends of the adapters with a fluorophore. So we can, after you transpose, you can actually image in situ to see which cells are accessible, which elements are accessible. You can then pick those cells and then sequence and find out what it is you've been imaging. Okay, so we're going to use this method to look at this question. So on the top here, again, I'm showing you the ATAC-seq track for the MYC-PVT1 locus. This amplicon encompasses those enhancers. So you can see high level of accessibility. At the bottom, now, I'm going to show you now these images. Again, it's a metaphase spread. On the bottom left, this big structure is a chromosome. These little dots are ecDNAs. Each one is a circle, as I showed you by EM. In the middle panel, now this is a DNA in situ hybridization for the MYC oncogene. You can see that the ecDNAs carry the MYC oncogene, these green dots. On the right-hand side is the ATAC-seq signal. This is the level of accessibility as reflected by fluorescence. Notice that this little ecDNA is as bright as almost this entire chromosome. Okay, so all the accessibility is actually on the ecDNA, which is quantified on the right here. So this really gives us a single molecule evidence that each extra chromosomal DNA is more accessible and therefore capable of supporting more transcription. So it turns out from further analysis that the ecDNAs are actually, they are chromatinized, they have nucleosomes and have histone marks, but the nucleosome compaction is actually different. ATAC-seq is a parent sequencing method. You need basically 2 transposition events for these primers to allow PCR. And so there's a fragment length distribution that reflect the chromosome based nucleosome compaction pattern. Essentially, if a nucleosome is sitting on top of a sequence, you can transpose to the left and right of it, but not on top of it. And so there's this footprint. So we can actually map out the location of the single nucleosome, the dimer, the trimer. And what we see is that the ecDNA lack the higher order compaction, and therefore the larger fragment length in these kind of nucleosome footprinting pattern compared to the chromosomal DNA. Okay. So what I've shown you then is that we can have chromosomal amplifications of oncogenes, but even more so on extrachromosomal DNAs, because each copy generates much more of the oncogene product. And this reflects really a new idea that an ecDNA has lower chromatin compaction, and therefore each molecule can support more transcription. This is, I believe, a new kind of epigenetic mechanism, because the same DNA sequence, when it's on a linear chromosome versus ecDNA, adopts different chromatin fates. And so why that choice gets made and has this altered gene regulation is going to be the focus of our discussion and ongoing research. All right. As we started studying ecDNAs, we started following their pattern, their behavior in both metaphase and interphase cells. And then we see something very interesting when we started doing DNA fish experiments. In this experiment, in collaboration with the lab of James Liu at Janelia HHMI, we are using a two-color fish looking at both MYC in green, which is part of the ecDNA amplicon, and also an adjacent DNA locus on chromosome 8. And so when you have a chromosomal chromosome, you basically see paired red and green dots like this, okay? And you have ecDNA alone, you just see green color. And so in the 7 copies of ecDNAs, we see this cloud of green. But moreover, I want you to appreciate that somehow all these 10 copies are clustered together spatially in the nucleus. We call this an extrachromosomal DNA hub, ecDNA hub. There are 3 cells in this field, and you can see that all 3 cells are doing exactly the same thing. They're ecDNA hubs whenever the ecDNAs arise. This phenomenon is actually, I think, more general when we look in additional cancer types. You hear prostate cancer cells, colon cancer, GBM, gastric cancer. We can see that using the same experimental strategy, multiple ecDNAs, MYC, EGFR, FGFR2. This is a line that has both two kinds of oncogenes on ecDNAs, MYC and FGFR2. They're all frequently seen in these ecDNA hubs. Okay, so this appears to be a general phenomenon. The hubs comprise 10 to 100 copies of ecDNAs. Their size are about 2 to 5, approximately a few microns, and they're quite prominent. They exist in interface. And we can actually quantify this mathematically. And so this is, we found that this autocorrelation score is very effective. This is essentially the probability, if you start from a pixel that has an ecDNA signal, what is the probability of seeing another ecDNA signal as you move away in distance from that index pixel? And so ecDNAs are clustered. Then when you walk away from that that initial index, you're very likely to see another signal, and then it decays over time. But if it's random, then when you walk away, you're as likely to see empty space as you are to get ecDNA signal. And so the gray line is basically the random control, and each of these, all 4 of these samples that we analyzed have a significant deviation, an increase in autocorrelation, indicating clustering behavior, presence of oncogene ecDNA hubs. All right. We, of course, wanted to verify that this behavior actually occurs in living cells and it's not just a feature of DNA fixation or fluorescent cytohybridization. And so we also made this TET-O knock-in array, knock into the MYC locus, ecDNAs, and then you can actually then tag them with GFP. And this movie in the center, you can see this is going to play for about 30 minutes. You can see this ecDNA hub is quite stable. It moves around and changes shape over time. But it's actually, it's not just a transient event. It's somehow a stable and dynamic structure. Okay, you can even see that the ecDNA hub is actually more compact, more tight in living cells. And that is because we think that to do fish, you have to do a little bit of denaturation, whereas in living cells, they're actually quite spatially compact. All right. We then next want to know, okay, great, we have the ecDNA hubs. So what is the relationship with transcription? So in this next experiment, this is a dual DNA and RNA fish. We're still following the MYC ecDNA chromosomal locus. Now on top of that, we also probe for the nascent MYC RNA by hybridizing the MYC intron. I hope you can see that this red color only shows up where the ecDNA hubs are clustering, but not near the chromosomal loci. And so from this kind of pair analysis, we can actually compute a probability of MYC transcription per DNA molecule. That's essentially what is the probability of catching this molecule of DNA in the active transcription at the time of the Fisher experiment. Okay, so it integrates both the bursting frequency and the length of time for transcription. So you can see then that on the ecDNAs, each molecule is more likely to transcribe than a chromosomal locus, significantly different. And moreover, we're really surprised to see this kind of cooperative behavior. Specifically, in each cell, the more copies of ecDNAs there are, each molecule is more likely to transcribe. And this actually specifically relates to the spatial proximity of ecDNAs shown on the right here. So we're looking at, on the x-axis, the clustering score. The more to the right, the more cluster it is. On the y-axis, the probability of MYC transcription. So the higher the probability of transcription, on the y-axis, the more likely to transcribe. You can see this positive and significant correlation. That is, the more molecules there are, especially when they're close to each other, each one of them is more likely to transcribe. Okay, so this is a very special kind of behavior that somehow occurs in these ecDNA hubs. Okay, we're really intrigued by these results. And so we have a few sort of key questions. And some of the questions that came up include, first of all, how does gene regulation operate in this ecDNA hub? I described this cooperative behavior that's sort of quite unusual. Second question, what might be the mechanism that would hold the ecDNA hub together? Is it some sort of force or some sort of protein? And thirdly, what might be the reason? Can we infer something for why these ecDNA hubs occur in cancer cells? What kind of selective advantage could they gain from this unusual sort of mode of behavior? Okay, so two very talented graduate students, King Hung and Katie Yost, took up these questions. And we first addressed the gene regulation question by thinking about enhancers, basically sequences that are needed for gene activation. And we decided to look at this question by comparing ecDNA-containing cells versus cells that have chromosome amplification. Our naive thinking was that you might imagine that the default idea is that you have ecDNAs, there are lots of copies, but their mechanism of gene regulation is exactly the same as the chromosomal version, but there's just more of it. So we can basically address that question by looking at an isogenic cell line for these colon cancer cells, COLO320-DM, that contain the MYC PBT1 ecDNA, versus an isogenic line where all the extra copies have reintegrated into the chromosome as a tandem amplification. And this isogenic line is called COLO320 HSR, okay? And so the cartoon on the bottom shows you what is going on with these oncogenes, and you're looking at metaphase spreads of basically, these are chromosomes in blue, glow green dots are ecDNAs versus reintegration in this HSR. And we're going to, and Katie and King used a single cell multiome method, which was developed in my lab and now commercialized by 10x Genomics, where we can actually track single cell RNA and chromatin accessibility in the same single cell. And what we thought we'd do is that we're basically going to profile tens of thousands of cells. And on the left, you can see that we actually did this on about 70,000 cells. And then from every, and this is on the bottom, on the left, we're showing this is a UMAP projection of a single cell. So every dot is a single cell. We can clearly distinguish the ecDNA containing DM cells in blue versus the HSR chromosome amplification cells in green. And we can also then see cells with high MYC RNA levels versus those with low MYC RNA levels. So what we thought here is that we're going to exploit the single cell variation. And because we know ecDNAs are heterogeneous and different cells carry different complements, we can think of this as a mutagenesis experiment of nature. We're going to select cells that have high MYC RNA level and ask what DNA regulatory elements that came along that supported the high levels of MYC transcription. So on the right, now I'm showing you basically cells been by MYC RNA levels high versus low, and also DNA regulatory elements that are showed up only in those cells. And we're looking at ecDNA containing cells in blue on the top versus HSR cells, chromosome amplicons on the bottom. And this analysis, of course, this is now MYC here, PBT1, identify actually 45 ecDNA specific enhancers that are associated with high MYC transcription at a single cell level. Okay, so far, so this analysis basically made the point that the same, so somehow different enhancers are engaged. This does not occur when you have high MYC expression in the HSR complex. They only occur in the ecDNA. So somehow more access to more enhancers in the ecDNA context. Next, King and Katie then actually conducted DNA sequencing, including single molecule nanopore long read sequencing to map out the ecDNA context. And here we got a real surprise because we thought that each ecDNA molecule must contain all the regulatory elements that it needs to maximize its expression. That's how it gives itself and the cell a selective advantage. Those of you who have worked with bacterial plasmids know that if you make an AMP-resistant plasmid, but you somehow screw up the cloning so that you can't express AMP, the next day, you don't have that plasmid anymore. You will lose that plasmid during the selective pressure because you can't express. But when we conducted these single molecule sequencing, we discovered really a high degree of heterogeneity in the ecDNA primary sequence. The second most abundant species was this 1.5 megabase species. There are 64 copies basically in these samples, and that comprise basically essentially this MYC PBT1 amplicon that basically looped around like this, okay? But even more abundant is a 86 kb, so much smaller ecDNA. That is essentially the first half of PBT1, so the PBT1 promoter plus the enhancers, then chopped off and delete the rest of the circle and fused back into the second exon of MYC, shown here. So this small circle essentially has the PBT1 promoter replacing the MYC promoter as a fusion transcript. It has captured the 3' enhancers, so the intragenic enhancers from PBT1. But to our surprise, is missing all the 5' enhancers of MYC, including all the ones that we showed in the single cell covariation is supposed to be important for MYC expression. So the surprise here then is that oncogenes are in pieces, and we have these ecDNA subspecies. When we now conduct RNA-seq of these samples, we discover that in fact, all the messenger RNA, 80% of the messenger RNAs are derived from this small ecDNA circle. It has this PBT1 basically as the 5' alternative start site fused to the second exon of MYC. Okay, that's the dominant oncogene mRNA template. That's what this circle is. So then where are those enhancers that are supposed to be driving this? They're on a different molecule. And in fact, we conduct a high chip experiment to show that the high chip showed that in fact this small circle is this very tight triangle right here. But there's extensive and promiscuous enhancer contact with this 5' landscape that comes from this second enhancer, from the second ecDNA circle. So in other words, we think that this is evidence for intermolecular transcription, enhancers from one ecDNA driving transcription from the second ecDNA. This small ecDNA is only an oncogene template, and this second ecDNA is only enhancers. Okay, it's divisional labor, specialization, and some sort of intermolecular cooperation, a real surprise. Let's think about then what has been accomplished by this unusual mechanism. First of all, is that this arrangement now allows this PBT1 promoter to access both the 3' and the 5' enhancer at the same time, which normally does not happen. You only have to choose one side or the other. Secondly, we have now permanently broken the PBT1 MYC promoter competition. The PBT1 promoter now permanently wins. It's now basically fused to the rest of the MYC oncogene, but now it gains all these enhancers from this intermolecular interaction. This is a really surprising result to us. And when we got this result, we thought that really the way to prove this mechanism is that we have to somehow break up these ecDNA hubs, because we imagine that the hub is necessary for this kind of intermolecular communication. And if that were true, we need to somehow understand the mechanism of the hub formation and the forces that hold the hub together. Now, we first thought that maybe this hub is just a function, a consequence of transcription, but that's in fact not the case. If we block transcription by adding alpha amanitin to block RNA polymerase II or 1,6-hexanediol that disperse certain kinds of condensates like mediator, these ecDNA hubs persist. You can see by the image and also by the autocorrelation graphs on the right. So we actually got a major clue about the mechanism from our earlier work on chromosomal regulation, the PVT1 locus. Sang-Woo had discovered that, in fact, BRD4 marks the winner of the promoter competition between PVT1 and MYC. And BRD4 is, of course, a reader of active enhancers. It mediates enhancer-promoter contact, long-range gene communication, and is a very interesting candidate for this idea. Additional studies, including classical work by Jay Bradner and many others, let me just remind you, found that, of course, BRD4 is a classic reader protein for the active enhancer mark HVK27 acetylation. BRD4 protein contains bromodomains, and that directly recognizes the acetylated mark. This is blocked by a small molecule JQ1 that displaces the bromodomain. And there's also an IDR, a disorder domain that can also drive, they can separately drive condensates. And this is on the right, I'm showing you an in vitro experiment published by the lab of Mike Rosen, showing that in the presence of acetylated chromatin, the bromodomain can drive this kind of phase separation and separate out different flavors of chromatin. Okay, so with the idea that potentially BRD4 as our mediator, here of this clustering, we went on and tagged the endogenous BRD4 by knocking in a halo tag. And then in our lifestyle image, you can see that in the center of every ecDNA hub, there's a BRD4 focus in the center, holding, hope you can see that they're holding the hubs together. And this is also verified by ChIP-seq experiments. So now on the bottom, I'm showing you ChIP-seq looking at both in the colo DM cells versus the chromosomal amplification. We can see that these enhancers that we have previously showed by single cell covariation are in fact marked by K27 acetylation, bound by BRD4, and also they have a TAC-seq. And we can also, and in fact, the PVT1 promoter has one of the strongest peaks for BRD4 occupancy. We also found something interesting, that is that there's a focal deletion of the PVT1 promoter in the chromosomal amplification in the HSR cells. So these two different lines have adopted different evolutionary strategies. And that is that in one case in ecDNA, this basically small circle that forms, PVT1 takes over the MYC promoter, drives MYC transcription. In the chromosomal amplification, HSR, a focal PVT1 promoter deletion to get rid of this tumor suppressor DNA element. Okay, so then the key experiments ask, can we then disperse ecDNA hubs by blocking BRD4? And that's this next image. So I'm showing you the 2D movies now, 3D movies. On the left is the control situation looking at the ecDNA hub. You can see that's really well formed. On the right-hand side, you can now see, sorry about that. On the right-hand side, I hope you can appreciate that in the presence of JQ1, all these ecDNAs are now dispersed into individual elements. Okay, so actually, I love this movie. I can watch it over and over again. Okay, and so in fact, we can show this is a very rapid process. This is lifestyle imaging. Now, in the presence of JQ1, we can see that actually as short as 20 minutes into adding JQ1 into the media, the ecDNA hubs start to break apart. They break into smaller hubs, and they finally completely disperse. So this is likely a direct effect of the BRD4 tethering. The consequence of breaking of the hubs are actually, I would say, pretty spectacular. On the left, we are looking at the MYC firing frequency. So a low dose of JQ1, the MYC transcription now dramatically reduces. This is specific to the ecDNA. On the chromosomal amplification, there's much lower rate of MYC transcription, and they're not affected by this low dose of JQ1, whereas the high cooperative transcription of MYC in the hubs are strongly affected. And there's a concurrent effect on basically killing cells that are containing the ecDNA, which is not seen, or at least somewhat selective, over the cells with chromosome amplification, and this is shown by the cell viability shown here. So I've shown you this idea of an ecDNA hub for the MYC oncogene, but we think this idea extends really to even other kinds of oncogenes and even different oncogenes interacting together. In the SNU16 gastric cancer cells, these cells contain two different oncogenes, MYC from chromosome 8 and FGFR2 from chromosome 10, that are both ecDNAs. When we do an interface, a metaphase spread, shown on the left here, you can see that the MYC shown in green and FGFR2 in red, they're actually separate molecules. But in the interface, which is caught in an adjacent cell, they actually both form hubs, and there's extensive intermixing, which is quantified on the right. That in the interface, about 50% of the ecDNAs are actually co-localized. Okay, so King and Katie wanted to show that in fact there's cross-regulation between the two different ecDNAs. We're now on this complicated figure on the top, on the x-axis. I'm showing you the MYC PVT1 locus in these cells. This is CHIPs to show that they're occupied by BRD4. These are enhancers marked by K27 acetylation. On the y-axis, I'm showing you now the FGFR2 locus, and they also have active enhancers bound by BRD4. In the center is a high-chip enhancer connectome map showing you the contact. In a subset of the molecules, our whole genome sequencing shows that there's a small translocation that's in a subset of molecules that serves as an internal control for a cis level high chip signal. That's this big red block in the center here. But in addition, there's a very strong and actually somewhat promiscuous trans contact between independent molecules that are intermolecular contact of these enhancers, two different ecDNAs. King went on to perform CRISPR-I experiments targeting all these enhancers across both different ecDNA amplicons. And so, and they are shown both in cis regulation in green and trans regulation in blue. So I'll just highlight the key points as if you silence the promoter of FGFR2 or promoter of MYC, you silence its own gene, but not the other oncogene, indicating that there's no trans regulation at the protein product level. However, if you silence enhancers, we can actually see evidence of trans regulation. For example, this intragenic enhancer FGFR2 locus, if we shut that down, we would then actually shut down MYC in trans, which is characterized here by this cartoon that in fact, in this presence of this hub, the FGFR2 enhancer is now influencing MYC transcription. Okay, so this is a form of combinatorial gene regulation in this dual oncogene ecDNA hub. In summary, what I've told you then is that, of course, you might expect that if we silence, if we add JQ1, we can silence both oncogenes. So irrespective of the product, both oncogenes are sensitive to JQ1 inhibition. All right. So in summary then, I believe that I've shown you that I think this ecDNA hub is really a really amazing kind of nuclear structure. It's a kind of emerging behavior of extrachromosomal DNA, like these kind of schools of fish swimming together. These ecDNAs in many molecules, tens to hundreds of molecules in the nucleus would actually get together and behave cooperatively. This is a unique kind of structure that is, I think, worth noticing, that we now understand the underlying sort of principle for their formation. In contrast to a chromosome, which is a long molecule DNA decorated by proteins, an ecDNA hub is actually a protein core, specifically BRD4, decorated by DNA. So the opposite configuration to a chromosome, which is why you notice that I didn't use the word double minute chromosomes, because in fact, the ecDNA hub is not a chromosome, right? It's a different kind of cytological structure. And the focus of the interaction is actually this bromodomain K27 acetylation interaction that can be broken by JQ1. So this is an ecDNA hub that exploits normal mechanism for long-range enhancer promoter contact that are exploited now, co-opted for ecDNA hub formation. This is we want to propose then that ecDNA hubs are actually the units of oncogene function and not individual ecDNA molecules, because only together do you get robust oncogene transcription. The gene expression in this hub is different. It's intermolecular. The enhancer promoter regulation can occur across molecules. It is cooperative. The more molecules are present in the hub, the more each one transcribes. And finally, it's combinatorial. Any enhancers that come into proximity into the hub, even from different oncogene loci, originally from different chromosomes, can actually now cross-regulate and drive transcription. I think functionally, then we can think that this has important implications for cancer. If we think about DNA damage and chemotherapy, that you try to treat cancer cells with it, these ecDNA hubs can tolerate high levels of mutation burden. In fact, I showed you that putatively suboptimal subspecies of ecDNAs can accumulate because they can just complement each other in the hub. But in conversely, if we understand this mechanism, if we break up this ecDNA hub platform, this actually could be effectively silence all the oncogenes on the ecDNA hubs, and therefore the hub should be a therapeutic target. All right, that is, I believe, the last slide. So I'm grateful to acknowledge many people involved in this work. The work on the TCGA was started by Ryan Corsens. The work on PVT1 and MYC on the chromosome was done by Sungwoo Cho and Jin Xu, both now faculty members in Asia. And the work on ecDNA hub was done by King Hung, Katie Yos, and Liang Qixi in the lab of James Liu and Bob Tijan at HHMI Janelia. I gratefully acknowledge my collaborators, Will Greenleaf, Paul Michel, and all my funding sources. Thank you for your attention. I will come back to this model slide and take some questions.
Thank you so much, Howard, for such a fantastic and interesting talk. So I'm going to start, there are a number of questions. So I'm going to start with the first question, which is, why is the ataxic DNA in the nucleus localized only in the periphery? And the person's referring to the images with MYC hybridized adapter DNA and DAPI.
I'm not sure if I know which image you're talking about, because I think if you're looking at the ataxy image, in the metaphase spread, the metaphase spread is done in a way that you actually break up the nucleus, so you can't see basically the boundaries of individual nuclei anymore. Is that what we're talking about? I hope that answers the question.
Sure. Question is from Sahil, and maybe Sahil might actually rephrase the question. So we'll move on to the next question, which is from Laurent Lecam. Great talk, Howard. If I remember well, those ecDNAs can be reintegrated into chromosomes at one point. I was wondering if JQ1 treatment favors such fusions with chromosomes by dislocating the ecDNA hubs that would be much more difficult to fuse.
Got it. Yeah, actually, I think that's a really interesting question. I think that in fact, the HSR cells are such an example of all the ecDNAs reintegrating into one spot. Our idea is actually that I think the ecDNA hub explains this very unusual behavior, because if you imagine there's 100 molecules of the ecDNA, and they also have to all independently jump into the same spot on the chromosome. It's very hard to imagine how that would happen, right? Each one after another jump in at this, into the same spot. They're much more likely to just randomly integrate over the place. But if the ecDNAs are all together, spatially close to each other in a hub already, then if there were a break, they might all integrate at the same time. I think I agree with your with the idea that maybe if you select against ecDNAs, you would favor hub formation. You would favor reintegration. You would basically, that would be a subset of cells that have HSRs that are in the population that will grow better. But I think on the cells, if they start with ecDNA, if you disperse the hubs, the integration might be more random over the chromosomes, as I described. We haven't done the experiment, so I don't have answers one way or the other, but I think it's an interesting thing to think about.
Thank you. The next question is from Yi-san. Great talk. So there are actually 3 questions. So if only the PVT1 promoter acts towards regulation of MYC, why is PVT1 link RNA essential in the CRISPR screen?
Right. So the link RNA, so there's 2 components. One is that the CRISPR screen is in the chromosomal context, and the PVT1 promoter was actually, it was not an essential hit, it was actually the opposite. The PVT1, basically the guide RNA silencing PVT1, basically knocking it down or deleting it makes the cell grow better. And that's why we led to this model of the competition between PVT1 and MYC, because whenever you shut down chromosomal PVT1, MYC will transcribe better. That was the reason. But in the ecDNA context, the cancer cells completely rewired the regulation so that PVT1 drives MYC expression instead. So that's a completely new and different strategy.
And another question from David Pellman is, can ecDNA enhancers activate genes on linear chromosomes?
Very interesting question. We have not looked at that question. I think the first question is going to be, what are the target genes of the ecDNA? Because the ecDNA is actually obviously floating everywhere. So unlike a normal enhancer on a chromosome, you can at least look at nearby genes. Here, we wouldn't even know which chromosomal genes are potentially its target. So it could be potentially broad, but I think it's interesting to think about that question. We have not done that analysis.
Okay. Next question from David Kierczewski. Can you assess ecDNA easily in TCGA and other large data sets and show a hypercorrelation between ecDNA and expression levels, for example, MYC?
Yes, so that was done in some of our some of the studies I referred to. We can tell that we have ecDNAs in TCGA and other clinical samples from two ways. One is that, as I mentioned, the ATAC-seq pattern looks quite different. You have this very high, sort of the contiguous, broad accessibility. And the second reason is that from whole genome sequencing, there are diagnostic, basically, some of these junctional reads. Basically, the graph looks like a circle, right? And so those two features together tell you that you're dealing with an ecDNA. And then indeed, when we correlate the RNA expression levels, basically those same samples have much higher level of the oncogene expression. And that was sort of the basis of that remark in the middle of my presentation.
Another question is, do you see just oncogenes in ecDNA hubs?
Do we only see oncogenes in ecDNA hubs? Well, the ecDNAs obviously often carry, basically, they're like, as I mentioned, like a megabase big. So they're obviously sometimes surrounding genes. And that's because regulatory elements are also dispersed. So many of those genes are carried along at the same time. So those are basically, you might think of them as passenger ecDNA cargoes. Those are the ones that we know about. Are there other genes? I think that it's possible, not that we know of, but I guess I can't exclude that.
Okay. And are there GWAS hits or SNPs linked to GWAS hits in the PVT1 promoter?
We did not see GWAS SNPs. Those are, in other words, inherited variants in the PVT1 promoter. Those are more in those PVT1 intragenic enhancers, as I was pointing out in the kidney cancer example. The PVT1 promoter is basically a hotspot for somatic mutations. So in other words, acquired mutations in cancer samples. A lot of structural rearrangements and deletions and inversions targeting that. And so there's a, yeah, that's what we know.
Then last question is, do you see non-coding RNAs or repetitive sequences like allos enriched in ecDNA hubs?
Good question. We did not, but I'm trying to think about whether we search for that. So in fact, a lot of those sequences, repetitive sequences, are associated with heterochromatin marks. And ecDNAs are depleted for heterochromatin marks. So if you stain for, let's say, H3K9 trimethylation and other features like that, they're essentially absent. So I don't think there's any evidence for those sequences being selected. I think that the questioner might be thinking that, oh, maybe those repetitive sequences allow recombination or some other event to generate the ecDNA. It's possible, but we don't see an enrichment of those sequences in the ecDNAs.
Okay, so that's all the time we have now for questions. And those of you whose questions were not answered in this forum, your questions will be emailed to Dr. Chan so that he can answer them directly. So finally, Howard, thank you so much for this wonderful talk. It was a great pleasure to have you. And thanks also everyone for tuning in to listen to this talk.
Thank you, everybody. Take care.
Bye.